Hi everyone, in this video we're going to talk about our lab on enzymes, nature's catalysts. So we're going to cover parts A, B, C, and D, and we're going to skip E and F. So in part A, we're actually going to prepare cheese. Well, sort of. So what we're going to do is mix milk that's been actually mixed with buttermilk, so it's been treated with buttermilk. And we're going to mix that with an enzyme called rennin. And this should produce curds and whey. So curds are the solid pieces shown in the picture on the left. Whey is the liquid portion. So rennin, again, is an enzyme that actually coagulates milk and turns it into more of a solid. It's a little bit of a wiggly solid, but a solid nonetheless. So we're going to produce curds and whey. Then we're going to separate out the curds using cheesecloth. So this essentially is just a filter. And then we're going to weigh the curds and see how many curds we produced, or the mass of curds that we produced. Now, at cheese factories, they actually take the curds, they let them dry further, they add salt and do some other things, and that's what leads to the cheese that we buy at the store. In part B, we're going to look at lipid digestion. So we're going to use an enzyme called lipase, although we're actually using a solution called pancreatin. So pancreatin contains lipase, and we're going to mix that with what's called litmus milk. So this is milk that just has some pH indicator in it. So litmus is just referring to that pH indicator. Now, the reason that we have pH indicator in the milk is we want to track whether our products are acidic or basic, and that will tell us if a reaction occurred. So looking up above, we have something pretty similar to the fat that's found in milk. So let's just write fat here. And when we add lipase, that's going to break apart the fatty acid portion from the glycerol portion. So we saw this in lecture before. Now looking at our fatty acid, notice that there's this carboxylic acid group. So as we start to break down the fat in milk using lipase, that's going to produce acidic molecules, those fatty acids. So the more acid we have in solution, that means we're going to decrease the pH because it's becoming acidic, and litmus will turn pink in acidic solutions. So what we should see is a pink solution at the end of the reaction, because that tells us that we are in fact breaking apart those milk fat molecules and creating fatty acids. In part C of the lab, we're going to look at how different enzymes may or may not break apart proteins. So these enzymes, or this class of enzymes, is called, or they are called proteases. And those are all shown down below. So what we're going to do is we're going to test the effect of each of these enzymes on gelatin. So gelatin is a protein that comes from collagen, and it can be hydrolyzed or broken down by pepsin and trypsin. However, it does sort of depend on the pH and things like temperature, etc. So we'll see what happens in this experiment. But pepsin is an enzyme that is in our stomachs, and it breaks down proteins into what are called polypeptides. Trypsin is also a digestive enzyme. However, this one is found in the small intestine, and it also hydrolyzes proteins. And then meat tenderizer contains bromelain, which is a protease that's found in pineapples. So often on gelatin packages, it tells you not to use any pineapple juice, for instance, because the bromelain will actually break down the gelatin and it won't form the jello-like structure that you're looking for. So we'll see how these react under certain conditions. And then in part D, we're going to look at how an enzyme can break apart sucrose. So sucrose, remember, is a disaccharide. So that means it's composed of two monosaccharides that are linked together. And those two monosaccharides are glucose and fructose. So what we'll do is we're actually going to mix sucrose with yeast enzymes. So the sucrose disaccharide will eventually connect with the active site of our yeast enzymes, and those enzymes should break apart the glucose and fructose monosaccharides, and then they'll be released as products. Now, to test that the enzymes actually broke sucrose apart, we're going to use Benedict's reagent. Now, remember, Benedict's reagent oxidizes monosaccharides like glucose and fructose, which are reducing sugars. So down below, I have a familiar picture that we've seen before. So Benedict's reagent is normally blue on its own, but if it reacts with a reducing sugar, it will turn orange or red, especially if there are large amounts of that reducing sugar. All right, so there's kind of two parts to part D. We'll first break apart sucrose into glucose and fructose, and then we'll add Benedict's reagent to see if we did in fact produce glucose and fructose. All right, so let's do an example calculation for part A of the lab where we created curds and whey. So in your lab assignment, you'll be asked to calculate a percent yield for this reaction. And that's going to be equal to the mass of curds that were produced divided by the mass of milk that we started with. And then we'll multiply that by 100. Now, in part A, we're not weighing the milk out, we're actually measuring the volume of milk. So we should have about 20 milliliters of milk at the beginning. Now we need to convert that into a mass. So what we're going to do is assume that the density of our milk is equal to 1 gram over 1 milliliter. So this should be a pretty easy conversion. We're just going to convert 20 milliliters of milk into grams by multiplying 20 milliliters of milk by the density, which is 1 gram per 1 milliliter. So milliliters will cancel, and we're just multiplying 20 by 1, so that will give us 20 grams of milk. Okay, so now let's say at the very end of lab we figure out that we had 3.290 grams of curds. So now we're just going to plug that into our equation up above, and then we're also going to plug in the 20 grams of milk. So then the percent yield will equal 3.290 grams of curds over 20 grams of milk times 100. And let's see, if we plug that into our calculators, that should give us about 16.5%. So this is kind of a low yield, but maybe something went wrong in lab, or maybe we squeezed a little too hard and some of the curds went through the cheesecloth. That can happen. All right. So hopefully this helps you with the lab, just kind of helps you understand what's going on during each part of the lab. And then hopefully the example calculation helps you on your post-lab assignment. But just let me know if you have any questions on Canvas.